Hello, Sonia. I am sorry to miss you today. I had some travel issues and ended up turning around and coming back home so that I wouldn't get stuck in Beijing. But anyway, I suggested to Gawei that I could participate remotely, and so here I am. So today I'll talk to you a bit about predicting nucleic acid binding sites on proteins. And we'll do this using some machine learning models. So I'm very interested in the intersection of biophysics and data science and try to build predictive models by combining calculations that are familiar to us as people in the biophysics community, but to use them in a way that is more along the lines of how we do things in data science. So here you can see a pair of molecules that are interacting, and so you can see that there's a binding site that we've marked in blue and a non-binding site that we've marked in red. Now, in this case, I'm showing you an example where the second molecule is another protein, but today we're actually going to talk about predicting these binding sites for DNA and RNA. And so the way we're going to do that is to come up with a large number of features. So in the table here, you can see that the rows are very are different types of features, and here the columns each represent a single amino acid that's been colored either red in the case where it's a non-binding residue and blue in the case where it's a binding residue. And so the idea is to find patterns and correlations in this matrix, right, that would allow us to distinguish in general cases the difference between blue points and red points. So to do this, the particular model that I'll show you has been built using support vector machines. And so, support vector machines basically map data up into a high-dimensional space. So you have some axes. Here I've just, given a two-dimensional example. So X1 is some type of a feature, something that was the row in our previous slide, and X2 is a different feature. So maybe X1 has something to do with electrostatics, and X2 has something to do with hydrogen bonding, and then maybe there can be other features, right? So all of this happens up in a very high dimensional space. And so the idea behind support vector machines is to figure out how to divide, right? So here we have black and white points instead of blue and red points, but same difference, right? So we're trying to figure out in some high dimensional space how to draw hyperplanes that are going to separate, right, the two classes, binder or non-binder. So in this example, you can see that this green line, H3, does not divide the two classes at all. But both the blue line and the red line do. And so which one is better? Well, most people can look at this and they'll say, well, H2 does a better job of dividing the space, right? Separating the black points from the white points. And the mathematical way in which we describe that is that the line, right, that H2 maximizes the distance between the boundary and the nearest data points, right? So here you can see that the distance between the nearest data point and the blue line, H1, is very small, right? But it's much better if we use the line H2, right? So that we can maximize the distance between the boundary and the data points, and this allows us to build a model that is, say, less ambiguous, right? So we have a large margin, right? We're enforcing sort of a large margin between where we're drawing the boundary and the nearest data points. Okay, so that's simple enough, and that's basically how linear support vector machines work. And then you can also develop nonlinear support vector models. For example, and usually this is done using kernel mapping. So here we're back to, blue and red, but suppose that the blue points again are binding residues and our red points are non-binding residues, but they're organized, right, in this way where it's going to be hard to separate them with a line. And so how do you draw other types of boundaries? Well, we do this using kernels, and so we take, in this example, we take our two-dimensional space and we map it up a dimension, right, in such a way, so we could use a quadratic function or a Gaussian function. So these are often called radial basis functions. And so once we've mapped it, right, up into a higher dimensional space, then we're actually able to draw a hyperplane that will separate these, right? And so drawing a hyperplane in this three-dimensional space is basically about, the same thing as drawing a circle, right, back in our two-dimensional space if we map it back. And basically I'm drawing This is usually done using kernel mappings. So for example, suppose that our data actually looked like this. So now we're back to blue points and say these are binding residues, and red points and these are non-binding residues. And we'd like to separate them, but unfortunately it's not possible to do this with a hyperplane, with a line. And so instead what we can do is map the data up into a higher dimensional space, and then, using something like a Gaussian function. So this is often referred to as a radial basis function. And you can see that if we map the data up into the higher dimensional space, suddenly it becomes possible to separate the data using a linear hyperplane. Right, so we can go ahead and separate the data. And in terms of how this looks in our original space, and when mapped back down to the original space, this is equivalent to dividing the data using a circle, right? And so the method then has figured out how to draw a circle instead of a line by mapping the data up into a higher dimensional space, and then splitting it with a hyperplane this way. Now that we know what a machine learning classification model does, let's talk about how one creates it. So first of all, we need some type of a training data set. So we need what's called labeled data, which means that we need a bunch of examples for which we have amino acids that are either binding residues or non-binding residues, right? And we need some features, which I'll talk about in some more detail. And then there's a process of feature selection and model optimization. So maybe I won't go into too much detail about how that does. You have to cross-validate so that you don't overfit your model. So that involves dividing your data, say, up into 10 chunks, and you train the data on, 90% of the data, and then you test it on the remaining 10%. And then you iterate over the entire data set, overall, say 10 of the cross-validation folds, in order to come up with an unbiased assessment of the model quality on the training data. And then you need some criteria for success. So sensitivity is the same as the true positive rate, specificity is the true negative rate. And precision is the fraction of things that we've predicted as the positive class that are in fact in the positive class. So if I predict 100 things to be binding residues and only 56 of them are, then my precision is 0.56. Okay? So these are all good metrics to look at when you want to assess the quality of your model. Accuracy, on the other hand, is not a very good metric for many types of problems. So for example, in the present case, if only 15%, within my data set, if only 15% of examples are true positives, right, then are positive examples, are binding residues, then I can get an 85% accurate model that has no value whatsoever. I just simply predict everything to be in the negative class, and that's going to be 85% accurate. So in general, accuracy is not such a good score. Now, F1 score is a very common metric if you want. So over here, we're talking, we're looking at true positive rate, true negative rate, right? Various things. But if you want one single number that's, that's trying to measure the quality of the model, right? So something to optimize essentially, the F1 score is commonly used. And so it's the harmonic mean of sensitivity, which is sometimes called recall, and precision. Okay, so that's a good metric that you can use if you're trying to optimize your model, and so on. Some people also like Matthew's correlation coefficient, but to each their own. Now, for training data, we took 262 protein DNA complexes. So these are derived from a paper on a method called DISPLAR, which is sort of the closest prior art that we found in order to do comparisons with an existing model. In their training data set, one protein RNA complex snuck in there, but we decided just to go ahead and leave it. This gives us over 50,000 data points, and around 18% are positive examples. So 18% are binding residues, and the remaining 82% are non-binding residues. So we created what are called stratified subsets. So that means that we maintain the ratio of 18% positive examples and 82% negative examples in subsets of the data. So we used 1,000 examples just for feature selection, 6,000 examples for some parameter optimization, because it becomes very expensive to do calculations on 50,000 data points. So you want to get some idea of where to look up in your feature space for good models, and you can do this using some smaller subsets. And then for cross-validation groups, in addition to dividing the data up, we also wanted to make sure that we were not creating bias in our cross-validation. In particular, related examples were put in the same cross-validation fold so that they would either be included all at once or excluded all at once. What you don't want is to organize your training data into folds in such a way that you have too much information in one fold that helps you predict in the other fold. So you really want to do this as independently as possible. Okay. Now for predictive features, we used a number of structure-based features, and so my group, kind of part of our art is trying to use our knowledge of biophysics to come up with features that we think will have predictive value in solving different types of problems. So some of our features have to do with hydrogen bonding, hydrophobicity, different things about packing density and plasticity. So plasticity, we developed an implicit measure for how deformable a particular site is. So can it rearrange itself in response to a binding event or something like this? And it's a fairly cheap calculation, but it's as much cheaper, for example, than doing a molecular dynamics simulation in order to figure out which parts of the molecule are more flexible than others. We also look at local microenvironments. And so I'll talk a little bit, given the interest of the group, I'll talk a little bit about electrostatics. You can also, do some microenvironments where you just do simple types of feature calculations where you say, how many leucines are within 5 angstroms of the amino acid that I'm making a prediction on. You can also look at secondary structure microenvironments, how much of my neighborhood is a part of a beta sheet. And also shape microenvironments as well. We also looked at some banded features, and so we looked at microenvironments that were not within, say, 5 angstroms or 6 angstroms of our amino acid, but maybe were a little bit more distant. And the idea was to to see if we could find some bandedness, right? So basically, if we could find like kind of stripes of positive potential or something, that kind of matched how DNA was laying down on the protein surface. And then in addition, we used just some evolutionary data, so information collected from close homologs of the protein. Okay. Now for the electrostatic features, so these are all derived from Poisson-Boltzmann electrostatic grid. So you could use any electrostatic method that you wanted, but we used PBEQ because it had a nice interface for us to do the calculations, and everything was calculated out on a 1/2 angstrom grid. Okay, and so what did we actually do? So now we have all the information. It's 3D spatial information on a grid, and what we really want is a residue level feature. So this guy right here looks like arginine, right? So we have this arginine residue. And what we would like to do is collect information from the 3D grid and map it to a feature for this arginine. And so we all know that if you look at the electrostatic potential mapped onto a molecular surface, it's very patchy, right? Inside the molecule, it has positive poles and negative poles. So how do we collect information about the electrostatic environment that another molecule sees as it comes in to make contact with this arginine? And so this was a feature that we developed. And so what we did was take information that came from annular environments near the surface of the protein, but not intersecting the protein interior. So you don't want to get inside here because, of course, the values are extremely large, have extremely large magnitude, right? So what you'd really like to do is collect information from the region out here, which is actually what the DNA or RNA sees as it's about to interact with, right, with our protein. And so, for example, you could collect information in an annulus that lies between the solvent excluded and solvent accessible surface. However, in our experiments, we found that it was actually advantageous to step the annular region back a little bit from the molecular surface so you could capture a softer electrostatic environment, right? So not quite so large in magnitude. So we collected information from here. If we trained models just on a single feature, obviously this isn't anywhere near the final model we got by combining information from multiple features, but if we just combined information from the electrostatic features alone, we could get, so this particular feature has no offset, so it's right up against the molecular surface. And its true positive rate was very low. If it did predict something binds DNA or RNA, then it did so with very high specificity, but overall this is not a very good model. Okay, same thing if we backed that annular region up 0.1 angstroms from the molecular surface, right? Really no change. But as we started to pull the annular region back from the molecular surface a bit, right? So increasing this void between the molecular surface and our annulus, you can see that we started to get, somewhat reasonable models. We could pick out, say, 33% of all the residues just by the electrostatic signature alone, right? And have pretty good specificity. Okay, so that's as good as we can do, say, with just one electrostatic feature. Now, after training our model with all of the features, we can go and assess performance. So here we can see that, on our training data set with the cross-validation, our true positive rate was 70%, and the true negative rate was 85%. So this is a good model. In addition, we had a test data set in which contained examples that were not used in the training data set, were somewhat independent from them. And here you can see that the performance holds up. In fact, on the test data set, the performance was actually a little bit better than it was on the cross-validation training set. So that was good. And then also, in addition to these metrics, another metric that we use to assess performance is the area under the ROC curve. So those of you who are not familiar with the ROC curve, I can draw a little picture to show you what it is. So normally we have a classification model, and positive scores correspond to positive predictions, and negative scores to negative predictions. So normally we cut off the boundary of scoring at 0. However, we can play a little game if we want. So we can say, okay, well, let's look at the distribution of scores for the positive prediction, or for the positive example. So the actual positive examples in our test data set. And so some of them will have been correctly predicted, right? And then if we have a positive example with a negative score, that is an incorrect prediction. Okay, so we have this histogram of scores for the blue points, the positive examples, and similarly we have a histogram of scores for the negative examples. Okay, and here you can see, right, so I'll go ahead and fill in, right, so the incorrect predictions for the red examples are these ones that I'm filling in. And the incorrect ones for the positive examples, I'm filling in here in blue, right? So, these are our false positives, these are our false negatives. Okay. So what an ROC curve does is looks at what happens if I take this score window and this score cutoff, and I start over here at one end. So here I have 100% sensitivity and I have 0% specificity, right? So I've gotten all the positive examples, but I've also predicted all the negative examples as positive examples. Now as I move it over, and so you want to picture I'm at one end here. Now as I move that score cutoff, right, I'm going to get some distribution of true positives and distribution of true negatives, right? And I'm going to keep measuring that and plotting it along this curve until my score cutoff has been moved to the other end here, where I have 100% specificity but 0% sensitivity, right? And so the curve is basically going from one corner to the other. And the more the score is able to separate the two classes, if I had them perfectly separated, so I had a peak over here, say -1, and a peak over here at +1, the graph would go straight up and over, right? And if I had a very bad model, it might just cruise along a line here. But the degree to which, right, we've optimized the area under this curve tells you how well the score is at separating the two classes. Okay, here are just some quick comparisons with other methods. So the most similar model that we were aware of for for this type of calculation was Displar, and you can see by comparison, DIBC has a much higher true positive rate, much higher sensitivity, and the specificity is about the same. Now, all the methods down here, bind, end, DBS, pred, and so on, these are sequence-based methods, and you can see that the sensitivity drops off quite a lot. So there is good information in the structure that allows us to make better predictions. Let's see, we also did some calculations where we looked at performance based on how similar certain examples were to something in the training data. So if we had very little sequence homology to an example in the training data, the performance still held up pretty well. If you remember, so this is on the independent test data, but this number right here, 0.68, is very comparable to what the cross-validation results for the training data were. And then just depending on the examples you pick, you might end up with a slightly higher sensitivity or lower sensitivity. But this is showing that the performance is not overly dependent on whether or not there's a related example in the training data. Okay, again, some more comparisons on independent data. Maybe in the interest of time, I'll go ahead and skip those. And then we also did some additional tests after the paper was published to take a look at things like conformational change. And so, for proteins that had secondary structure changes, order to disorder transitions, and so on, we looked at 79 different examples and we kind of tabulated up the results. And in fact, the results were actually better for this group of proteins than even for our test, our independent test data and so on. So we got almost 80% true positive rate. So recall is the same as sensitivity. And the specificity was extremely high. And so even on more challenging cases in which there is a non-trivial conformational change, the method seems to do well. Okay, and we even looked at some disordered examples. So these are ones in which we have a disorder to order transition. Now obviously they can't be too disordered because we have to have crystal structures for the disordered example. It just means that there's a really significant conformational change, something where secondary structure only happens upon binding the DNA or RNA. And so we were able to find 11 cases where we had PDB files for both the ordered and disordered regions. And we did calculations on both. So, we did DIBC predictions on the ordered examples and disordered examples, and we looked to make sure that, that these were consistent. So we only counted, say, a true positive if it were predicted in both the disordered and ordered examples, and only a true negative if it were predicted in both of them. And so again, you can see that the method did really quite well. So it got 80% recall or sensitivity, and again 97% specificity. So again, really good, even better than the training. Okay, maybe I'll skip examples. So heparin is related chemically to the phosphate backbone of DNA, so we use particular application. This is an example where you have a protein with a transcription factor fold that doesn't appear to bind DNA, and the prediction difference in prediction results definitely shows that. So there's sort of no prediction of a DNA binding site on this CoQ9 protein, whereas a FATR transcription factor with this exact same fold basically lights up for binding DNA. Okay, and finally, how about RNA? So RNA shares some chemical similarities with DNA, obviously. It's a little bit more complicated because we have, single-stranded RNA, double-stranded RNA. But we went ahead and did some calculations, right, to see whether or not DIBC would predict the binding site for RNA, both single-stranded examples and double-stranded examples. And it actually did better than we expected, but we're in the process of retraining the model to perform best on RNA, but it maintained its high specificity. So if it predicted something bound RNA, it was very likely to bind RNA. Now, compared to DIBC on DNA binding proteins, the recall or sensitivity was lower. So instead of predicting, 70, 75, 80% of examples, it was predicting, just for this 19, the set of 19, it had a sensitivity of 42%. Now we did a much larger test. So over 200 non-redundant examples of RNA binding proteins. And so, the specificity continued to be high, so 0.85. The sensitivity on that data set was a little bit higher than the previous 19 that I showed you, but this is a much larger data set, and so this is a better metric. So 55%. And the area under the RUC curve was really not that bad. 0.79 is actually a pretty good AUC score. So the model already has some signal, but we're already in the process of re-optimizing it to do best on RNA. So with that, I'll start wrapping everything up and also remind you to try our website. So I'll give you the website address in a moment. But here you can see that you can, upload your protein and it'll make predictions for you. So red is predicted to bind DNA. Anything in blue is predicted not to bind DNA, which is sort of opposite the electrostatic convention, but don't let that fool you. So anyway, the website has a little visualizer, and you can go ahead and play around and even make some pictures if you would like to show DNA or RNA binding predictions on your particular protein. So conclusions, DIBC works well, and it even works pretty well on RNA, but we're going to make it better. If you want to try it out, it's dibc.mitchell-lab.org. Funding for this is from the National Science Foundation. And the original DIBC model came from Xiaolei Zhu and Spencer Erickson working with me. Shravan Sukumar has helped out, with the website and testing it on order to disorder transition and other examples. And Jack Gellerman and Michael Hermsen have been doing the work on RNA, and we're just about to start writing up the paper on those results. Thank you very much, and I'll be happy to take any questions. Well, I don't hear any questions, so I guess I'll just thank you again and have a lovely, lovely time in Sanya without me. Bye-bye.